So I'm very happy to be here, and I'm grateful to Stan for this invitation and to everybody for being here. I want to comment or raise the question of how we human beings came out of this original mess on the fifth day of creation. And it was not certainly with a throw of dice, because we all know that God doesn't like to play dice. But he or she, we don't really know whether it's a he or a she, let's say she, likes to play many other games. For example, she picked this mammalian card out of several vertebrate classes that could have been chosen. And later, among several mammalian lineages, the ball fell on the Homo sapiens clade. So what I would like to say is that these two games, the three-card game and the roulette, are in fact very different games. And I try to say that with this cartoon, where I put in very loose terms structure and function. And the first game is essentially how mammals diverged from other amniotes. And I put that in this vertical arrangement. The second game is how we humans got differentiated from other mammals. And the idea that I would like to suggest is that these are two very different types of phase transitions, in that in the first case, there is a qualitative jump in the structure of the brain with continuity in function. And in the second case, there is instead a qualitative jump in function with continuity in structure. So that's what we are going to kind of talk about. Yasser already introduced this fascinating structure inside the brain of mammals, the hippocampus, that you see here in different species. The exact geometry where it's positioned in the brain varies a lot. But in fact, it's not that new, the hippocampus, in that, as Yasser said, you can find it in reptilians, in modern reptilians. And it corresponds to this region of the medial pallium, the medial part of the dorsal aspect of the forebrain. So we can say it has been around for maybe over 300 million years. But then in mammals, it acquires a new internal organization with these three main subfields that you may have seen the acronyms of. And this has been remarkably preserved. So you can put pictures from different mammalian species on top of each other, and you don't have to move the labels of these three regions, dentagyrus, CA3, and CA1, including the hippocampus of a human brain. So the mammalian hippocampus is different from the reptilian hippocampus, and is different from the hippocampus of birds that we don't go into, that has also a more structured organization, but different from the one of mammals. At the core of the hippocampus of mammals, there is the dentagyrus. And modern genetics tells us that there are a few genes that regulate the development of different parts of the forebrain in particular. And although there is not a single gene that is responsible really for the dentagyrus, still by manipulating by producing mutants for a few of these genes, is possible to create mice without the dentagyrus. Unfortunately, these mice also lack other things like the urogenital tract, and maybe because of that they die a few hours after birth, so it's not really possible to test what Yasser told us, spatial memory function in these mammals without a dentagyrus. But nevertheless, we can get from genetics the suggestion that this insertion of the dentagyrus into the homologue structure that was already present as medial pallium in pre-mammalian ancestors was a kind of phase transition, was the all or none, essentially turning on of a new pattern of gene expression that produced this structure that is simply not there in this component of the hippocampal structure that is not there in modern reptilians. So it's a kind of qualitative change in the structure. And there is one species, the opossum, maybe it's not the only one, it's a kind of mammal who is a bit uncertain whether to be a mammal or not. So if you take cross-section of the forebrain from more rostral to more caudal levels, you find that the rostral part, there is essentially no dentagyrus, no mammalian hippocampus. It is like a reptilian forebrain, and then at the back you see a full-fledged mammal. So this is one of the elements that suggests that this qualitative change in the structure is not something that endows the animal with, or the species with a radically new function. So maybe it's a new idea not for philosophers but for engineers, in the sense that maybe what it buys the species is some kind of quantitative improvement or improvement in the fitness function, in doing things that it was doing already before. And this idea derives strong support by this beautiful and kind of not very well-known set of experiments by a group in Spain, where they have extended the classic, one of the classic experiments of hippocampal science, testing spatial memory in a water maze, to a number of non-mammalian species, including reptiles, birds, but even fish. Fish do not have a cortex, and therefore their pallium is completely different. And to make matters worse, the region that in mammals and in other amniotes is the hippocampus comes to be at the medial surface of the two hemispheres through this process of evagination. Fish forgot to go through evagination. They have another process called eversion, whereby the homologue structure comes to be at the lateral side of the forebrain. And yet, if you lesion that part that is homologue to the hippocampus of a mammal, of a reptilian, of a bird, if you lesion it in fish, you have problems in spatial memory that you can test with a fish analog of the water maze, which is really a very elegant experiment. You would think that fish like to swim, that's what they normally do. But this group in Spain managed to construct an experiment in which they motivate fish to get out of a certain region of the pool, and they can show that if you lesion this part of the fish forebrain, they have problems remembering where is the escape route from that portion of the pool. So in this table, it's a summary of several studies where they can say that the things that for all purposes, we associate the hippocampus to, in mammals, cognitive mapping and place learning, are also associated with the homologue structure, the hippocampus of reptiles, birds, and teleos fish, even though maybe we should not call it hippocampus, but it's the homologue structure. So there's strong support for the idea of continuity in function in the face of discontinuity in the structure. And therefore, we should also aim to understand why was it good for us mammals to develop an hippocampus with that specific internal organization with the dentate gyrus, if it was not a way to acquire a new repertoire of functions. And I'll just show you a slide on the attempts we have made in my group to understand that. This is with the student Erika Cerasti. So this is a diagram of the internal organization of the hippocampus. You have these three main regions and another region called the subiculum. And the set of connections, there are connections that come from the cortex. And there is this most interesting set of connections called the mossy fibers that link the first region in a sort of information flow through the hippocampus. The first region is the dentate gyrus with the second region called CA3. And what makes these connections so interesting is that these two regions, the dentate gyrus and CA3, receive the very same fibers coming from the cortex here. So they are exposed to the same sources of information. And the dentate gyrus sends its output, whatever is the computation that it performs. The result of this computation is sent to CA3, which already has available the input that was available to the dentate gyrus. So it is as if I speak to you, and at the same time, there is this gentleman who translates what I say in some other language and tells you the same thing simultaneously. Now, it would make sense if you don't understand what I'm saying in my language. But why should CA3 not be able to understand what the cortex tells it? It's an interesting riddle, this emergence of the dentate gyrus in the mammal, because it is not that the dentate gyrus does something else that then is sent to another part of the brain. So with Erika, we considered a model which includes the other projections that I don't spend time on. And I just show you this plot which has the result of simulations and analytical result. And first, you should see the quality of our work is that we get an unexplained factor of 4 between the simulations and the analytical result. But both show a maximum in this quantity, which is a measure of the information content of one of these spatial representations stored in the CA3 region, if the CA3 region listens also to what the dentate gyrus tells it, with one parameter, which is the number of synapses onto a CA3 cells that come from the dentate gyrus. So the other extraordinary bit of circuitry here is that the cells in CA3 receive thousands of synapses from the other two sources, the cortical projections and the projections from the other cells in CA3 that make these recurrent connections. So there are thousands of synapses onto each cell of CA3 that come from these other two sources. But there are only tens of synapses from the dentate gyrus. So this new twist in the organization of the hippocampus that only mammals have it's strange because it's a repetition of what the cortex already tells to CA3, but it's doubly strange because this repetition is conveyed by very few synapses. And in this riddle, which we still don't really understand, but it is intriguing that in our calculation, which could be all wrong, however, we get a maximum for the information stored in a CA3 representation when the dentate gyrus communicates with very few synapses, in the order of tens of synapses to CA3. So the details are not important here, but the general point that I want to convey is that if there is something in this model that is correct, it suggests that the insertion of an entirely new piece of tissue produces a quantitative improvement in this function of spatial memory that the hippocampus was doing also without the dentate gyrus. So that's for the first game, the differentiation of mammals from other amniotes. And I want to spend the rest of my time commenting on this other differentiation of the human species from other mammals, in particular in relation to language. And the first commentator on this problem is King Solomon. In one of the pieces of the Ecclesiastes that are more difficult to translate, he seems to say that God gave language to humans just to show how similar they are to bees. Probably meant other mammals with bees. This statement is maybe paradoxical and is of no comfort to other mammals who yet do not understand why they cannot speak. But a person that claims to have resolved this enigma is Noam Chomsky, who stated that monkeys cannot speak because they lack a language acquisition device, this LED, which is endowed a priori with a universal grammar. And the notion of this LAD, the language acquisition device, is really at odds with what neuroanatomists have seen in the human brain, that is no extra language organ, nothing really in the human brain, even in terms of subdivisions of the frontal lobes, that makes it different from, say, at least the brain of other primates. Then Chomsky changes a bit his position and says it's not really a device that monkeys are lacking, but something more ephemeral, a process, something like an algorithm, something that goes on inside the same brain that primates have. And he calls this process infinite recursion. What this infinite recursion really is rather ill-defined, but there is a review in Science in 2002 with Hauser and Fitch, where they have this picture, which is the most precise way in which they define infinite recursion in relation to this idea that this is the uniquely human component of the faculty of language. So in this picture, there is the faculty of language FL, which can be intended in a broad sense. So there is a surround that includes components like a system for concepts and intentions, and the sensory motor organs to produce and receive language input. And these, they say, are specialized in the human species, but it's not this specialization that make language unique to humans. What is unique to humans is the faculty of language in the narrow sense, this process of recursion, that they depict it is like a cartoon, but is the theory they present. Infinite recursion is an algorithm by which elements from possibly a finite set. Here the elements are words, but they could be at a subword level phonemes, or at a superordinate level they could be phrases or sentences. So these elements are joined one to the other like in a puzzle. So the different shapes of the elements indicate different rules for making a choice of element n plus 1 when you have already a sequence of n elements. And potentially these sequences are infinite, of any length, but also of an infinite variety because of combinatorics. So in a nutshell, in this review, infinite recursion is a very simple process where there are some rules that may be unknown, may be implicit, and these rules combine elements from a finite set to generate an infinite sequence. Now, if that is unique to humans, it's interesting that neurophysiologists like Moshe Abeles, that record from the frontal cortex of monkeys, find ways to describe neural activity, the neural activity they observe through some kind of data analysis, through some computer algorithm that extracts from this activity a putative sequence of states that are linked in a succession, in a sequence, in the frontal cortex of a monkey that is thinking about how to execute a certain task. So it seems that something concrete, like a concatenation of quasi-discrete states, can be observed by neurophysiologists in monkeys, and therefore maybe it does not correspond to this process that Chomsky et al. Think is uniquely human, unless what is uniquely human is the infinite character, the fact that Chomsky, Hauser, and Fitch explicitly state that this ability is not limited to putting together 3 or 4 elements. They review experiments of attempts to teach language to other species, including monkeys, and they say there is no problem with making them understand symbols and meanings, the association, and making them concatenate symbols, but only up to a limited number, like 3 or 4. So maybe it's the infinite character of this recursion that is unique to humans. Maybe. But again, neuroanatomists like Valentino Breitenberg, dissecting brains of monkeys and humans, they find only quantitative differences. So Valentino Breitenberg was a very insightful neuroanatomist, and he's inspired, he's kind of had very inspirational insights, one of which is the basis for the little model that I want to kind of introduce in a few minutes, is this idea that you can take the cortex of mammalian species that differ by almost 4 orders of magnitude in the total number of cells contained in the cerebral cortex. He says, let's look just at the pyramidal cells, the principal cells, those that project outside of the local cortical patch. You can think of this system of n pyramidal cells, which across mammals, n varies by almost 4 orders of magnitude. You can think of them as being partitioned into square root of n compartments, or patches, these colored objects, each containing square root of n cells. And then the 2 main systems of synapses on these cells, those that are on the apical and basal dendrites, are prevailingly, it's not a strict rule, but prevailingly, the apical synapses on the apical dendrites are those that connect different compartments, and those on the basal dendrites are those that are inside a compartment. They are local. They are connections between units that are neighboring in the cortex. So if you assume that each of the square root of n cells in one compartment has basal synapses with each other of the cells in the same compartment, and as one synapse coming from one cell in each of the other compartments, then a typical pyramidal cell receives of the order of square root of n local connection and square root of n distant connections. And although these numbers are just kind of ballparks, Valentino Breitenberg claims that they give at least a first impression of the scaling laws going from very small mammals to mammals with a large brain. From 10 to the 6, 10 to the 10, you have a kind of at least a pictorial impression of how this connectivity is always a local connectivity, global and local at the same time. The additional claim of Valentino Breitenberg is that in terms of the way this connectivity operates, it makes sense, according to him, to think of both local and long-distance connections as implementing the kind of associative memory that can be modeled in the way that Yasser mentioned in the previous talk. So one way in which you can make this model of Breitenberg amenable to analysis with quasi-physical techniques is by taking these patches of cortex of the Breitenberg model and reducing each patch to a single variable, a Potts variable, a variable that can take one of a number of states. This would correspond to the idea that each patch is a kind of Hopfield network. I don't know if John is still here. So, inspired by the Hopfield model, we can think of each patch as a system that can take one of a number of local attractor states and then can be represented by a Potts variable. So in the end, you reduce this two-tier system, local and long-distance connection, to a system of interacting Potts variables. And this was a necessary step because we tried many years ago to analyze the full neural network, where you have units that would represent neurons that are in these patches, and then they have the short distance, long distance connections, and a global activity pattern would be a collection of several attractor states, each expressed in one of the patches. But this model can be analyzed only in its simplest format, where it's not really interesting for computational purposes. And when we introduce these modifications that make it more interesting neurobiologically, of having a long-range connectivity that is not really random but is structured, and having the possibility for patches not to participate in the representation of a global pattern of activity, so including a zero state in the Potts units, then we cannot analyze the full model, and we have to reduce it to the Potts model. So the Potts model is a way to handle analytically this kind of simplified version of the Breitenberg model of the cortex. So this Potts model is a version of an associative memory. If it has stored a number of items, objects in memory, then when it is exposed to some external input that is correlated with one of the memories, it has the power, even if the correlation is weak, to make the full memory state across all the cortical patches that participate in it emerge. It's, I would like you to think more as a kind of extended version of the Hopfield model. If you want, is related also to Canerva's ideas, but I think it's better to keep John's model in mind as a reference.
It has also symmetric connections?
So yes, you can analyze the model with symmetric connections, and then you can use a kind of energy functional. You can use the same trick, only with the complication of the Potts variables. It has, this in fact was a model analyzed by Ido Kanter in 1988 as a kind of interesting exercise. He was not particularly interested in the cortex, but he took it just as a well-defined mathematical model. And he found the interesting result that the number of memories that you can store in this kind of Potts version of the Hopfield model is something that scales linearly in the number of connection of each Potts unit. So each Potts unit is linked to other Potts units. So the connections between these Potts units represent long-distance connection in the cortex. And C is the parameter that quantifies how many there are of these long-distance connections. Although these connections are tensors, because connections between Potts units are tensors, so they're a bit more complicated. They're not really representing synaptic connections, only in a kind of effective way. It's linear in C, the capacity of the system, but it's quadratic in S. S is the number of local Potts states, of local attractor states of each patch. So that's an interesting property. It works as a high-capacity associative memory. But the other interesting property is if you simulate it, here the system was in the resting state, and then at some point it was given an input that was highly correlated with one of the memories, and it retrieved that memory immediately, in no time. You don't even see the upshoot in the trajectory of the system. What is plotted is the overlap with all the various memories. Most memories have a vanishing overlap, and one memory has a very high overlap. And here, in this version of the model, there is just one extra ingredient, that is adaptation. Adaptation in the simplest sense, as Adrienne was mentioning this morning, in terms of firing rate adaptation, although it could also be synaptic adaptation. So this adaptation makes the network slip away of its attractor state after a while. And sometimes this happens. There is nothing special about this simulation, nothing to write home about. But in other cases, when the network slips away of its attractor state, the same attractor state acts as a cue to retrieve another attractor state. And in turn, this other attractor state can act as a cue to retrieve a third one, and so on. And the process can go on for a few steps, and then activity dies out. And yet, in other cases, it can go on indefinitely. So this is a kind of spontaneous behavior of the network. It's not really spontaneous because we can simulate this network as a purely deterministic system. But essentially, these jumps from one state to the other are unpredictable if you have just assigned to the network these memory items with some macroscopic rule. That depends on very fine details over which typically you have no control as an experimenter or a simulator. So it's a kind of output that the network produces, and in which you can hope to see some characteristic of the structure of the network. So you can think of exploiting this so-called latching dynamics as a model that in this simplified model, the model contains no structure and the latching dynamics will not express anything interesting. But you can think of a model that has some structure and that maybe its quasi-spontaneous dynamics may become an interesting model for something like a train of thoughts. And I like here to bring up this cite from Thomas Hobbes that points out that the main characteristic of a train of thoughts is that not every thought to every thought succeeds indifferently. So a process like this is interesting if it is not completely random. And in fact, you can ask whether in our simplified Potts associative memory, this succession of steady states is random, or else is fixed, or else it's, in the interesting case, in between. And in fact, with Emilio Kropff years ago, we found that by regulating a very simple parameter, which is essentially just a threshold, by varying a threshold in a certain range, you can go more or less all the way from a deterministic process, which is not interesting, to a random process, which is not interesting either, but going in the middle region, which is the interesting one of maximum complexity, where the network follows some kind of structured transition probabilities that in this version of the model you have no control over this structure, it's implicit, but it's something that governs the spontaneous dynamics of the network. But the question I want briefly to kind of conclude with is the question of when it is that this dynamics proceeds indefinitely. Because this was the characteristic that, if we believe the kind of revised Chomsky story, is unique to humans. The idea of a process which is in some sense recursive and can be carried out indefinitely. So Eleanor, another student in the group, did a study of this character, and here it's simulations in which the network is loaded with more and more memory states. P is the number of memory states, and on the x-axis. On the y-axis is the duration of this latching sequence in a certain number of time steps. It doesn't matter what it is. You can see that when the network is loaded with few memories, its latching sequences are short, but then at some point they explode with the behavior which is reminding of a phase transition, as if there is some critical value, which we can call PL, the critical value for latching, above which the network starts to latch indefinitely, to produce kind of sentences that are indefinitely long. So we can understand this intuitively with these simple pictures that are similar to what Yasser presented. Attractor states as holes in some kind of putative free energy landscape. If the holes, the valleys, are sparse, when the network is in a valley, the adaptation is like the lifting of the bottom of the valley. But if the valleys are sparse, when the system is lifted out of its current valley, it does not go anywhere else. Activity dies on this blue surface. But if the states are closer to each other because there are more of them in the space in which they live, then the network can jump, as it comes out of a valley, can jump into another neighboring valley, and so on. And eventually, there are enough valleys that the system has a kind of percolation transition that it continues to jump into another valley without ever ending the process. So one might hope to describe this analytically if one has, as we were saying, a symmetric a connection with a kind of energy functional that would be simple extension of the Hopfield model energy function. But the presence, so one would have defined this Potts variable in a certain way and some dynamical equations. But the presence of the adapting thresholds, this adaptation, model of adaptation, makes the simple energy description not applicable. However, we realized with Eleonora something that maybe many other people know as a trivial fact, that you can describe a system of this kind with two functionals, an analog of the energy and something that we call the adaptation functional, that are functionals of the Potts variables and of the thresholds that are treated as parameters. And the way these two functionals behave is that when the network undergoes this latching dynamics, the energy is not strictly speaking a function that is bound to decrease, but it does decrease at the moments when there is a transition. So you can think of breaking the latching dynamics into long periods when the network is more or less in a stable state, and short periods when it has a transition. And in the limit, that the short periods are very short compared to the long periods, then during the transition, the energy decreases and the adaptation functionally increases. You can look just at the blue curves. And during the long periods, the energy catches up again, and the adaptation instead slowly decreases. So this seesaw dynamics offers in a limit, it's only valid in the limit of separation of timescales, a way to describe analytically this latching process. And with Eleonora we did that. We got some kind of phase diagram here in two variables that I don't want to spend the time to explain. This is an analog of a temperature, and this is a variable that, a parameter that enters this Potts description. You have several phases. When the network has no latching, it has latching but finite, and infinite latching, and then if this W parameter is even stronger, it goes back in a state where it does not latch. So the analysis and the simulations are more or less in agreement, and this was done in a simple case without correlations among the patterns, but you can extend it to a case with the correlations. I don't want to get into the detail there in a paper. But now, with this more or less analyzable model, we can go back to the kind of grand and maybe very silly speculation about the evolution of the human species with these diagrams. So here I plot, there are two diagrams, one in which the x-axis is this measure of long-range connectivity, the number of connections between that a Potts unit receives from all other units. And in the other diagram, the x-axis is the number of states of a Potts unit, which indirectly in this model would be a reflection of the local connectivity. The local patch has a number of attractor states that varies with the number of connections of its constituent neurons. So S is indirectly a measure of the local connectivity. And then on the y-axis of both diagrams, I have this semantic depth, which is the number of memories that I can load onto the network. So as an associative memory, this associative memory of Potts unit has a region where it works, which is the yellow region, a region which is delimited by this line, which is the standard phase transition that you have when you overload an associative memory. It's a line, it's a curve that is linear in C. So the critical value for the associative memory to work is a number of stored pattern that is linear in C. and this is essentially the Cantor result, is quadratic in S. But then there is other critical value for the latching process to occur. So first, I forgot to say, we can think that some of our ancestors had a cortex that, in terms of parameter, were somewhere in the yellow region, because they had a cortex that could function as an associative memory. And then there is this latching process, and I said that latching has some lower critical value in the number of patterns. Patterns have to be, the number of memories has to be high enough that the memories are close to each other for latching to proceed. And it's difficult to estimate this value, critical value PL, but you can kind of follow at least the hypothesis that this is a number that does not depend on the long-range connectivity, and it's linear in S, which is something that we get from simulations. Then if that is the case, in these two phase diagrams, there is this red region, which is the region where the cortex would work as an associative memory, but in addition, it shows this latching behavior. So it can produce these spontaneous or quasi-spontaneous sequences that maybe in a system that is less unstructured than this one, that has some kind of structure, can be endowed with some interesting meaning. So the thought in this evolutionary perspective is that maybe it was simply an increase in the long-range connectivity or in the local connectivity, which is reflected in the number of local attractor states, that brought our ancestor from having a standard mammalian cortex that works, according to Valentino Breitenberg, as an associative memory. The same system would be endowed with these new dynamics, which would correspond to the idea of infinite recursion. So infinite recursion may emerge through a kind of spontaneous transition induced by the increase in connectivity. Of course, to make sense of this hypothesis, one has to study more structured models, and I just put up this slide, but I don't want to get into that. It's something we are trying to do now, to kind of differentiate inside the cortex regions that would correspond more to what linguists call operators, and others that would correspond to what they call fillers. This is something that I don't want to discuss. I just want to conclude with this slide, where here we see that this is a plot in which there is time, millions of years ago, on the x-axis, and cranial capacity, a measure of maybe the number of neurons in the brain of our lineage, maybe also indirectly of the connectivity. But it's a very indirect measure. Luckily, there is this guy Elston, that has more directly measured cortical connectivity in terms of the number of spines on pyramidal cells of different species. Here he contrasts primates, like a human, a monkey, a marmoset, old and new world monkeys, and he finds that looking only at the spines are an indicator of the number of synapses from distinct input units to a pyramidal cells. And he only looks at those on the basal dendrites for various reasons. But he finds, first, the surprising result that in the occipital lobe, pyramidal cells have many, many fewer connections than in the temporal and frontal lobe, which is an interesting result in itself. But then that contrasting human with monkey and marmoset, that of course differ by much more by at least an order of magnitude in terms of number of units and or volume of the cortex, but they also differ by maybe around the factor of 2 between human and macaque in terms of the connectivity of the number of connections per pyramidal cell. So there is space in the kind of range of variation of this connectivity parameter as we go to the human species for having a phase transition, having the same system also produce another dynamical behavior that may correspond to this kind of idea of cognitive scientists of infinite recursion. Thank you very much. This is the kind of thought for the future.